So we are starting the second session of the workshop. Because Vlad is traveling right now, and we don't want to have any problems with the internet connection, I will play a pre-recorded video of his presentation, and after the presentation he will be available for any questions that you may have. Let me share the screen first. Sharing the screen and
surface between the.
Oops, I think we need to start it from the beginning. So now I'm starting the presentation for Vlad.
Hello, everybody. My name is Vlad Constantinescu. I am an assistant researcher with the Bioinformatics team, part of the System Biology of Aging group. And this will be a pre-recording presentation regarding the Bucharest Pile, which was our system of neural networks creating an embedding for multiple graph concepts, genes, gene ontology concepts, and so on, designed to provide an informed prior for an inference problem that we had throughout the project, which was that of determining the pathways which contribute to longevity. Now, although this will be a pre-recorded presentation, I will be available for questions I think at least on the very least on chat, because of intermittent connectivity there may be some issues, but please feel free to stop at any time and ask any kind of questions regarding this machine learning effort, and there will also be some Q&A time at the end of the project. So without further ado, let's move on. So let's start with the problem that we were looking at as we've started the Bioinformatics project, which was we've had some information about different expression levels of genes collected from microRNA assays between individuals in the wild and individuals which had some kind of an intervention for longevity that was significantly expanding their lifespan. These were mostly collected from the gene expression omnibus in the form of GSE modules and so on. And most of the focus was on 3 species because these were intensely studied for longevity, the C. elegans form, the mouse, and humans. And we were looking at setting up a prediction problem which would predict the log fold count of a particular gene between a wild type organism and an organism with an intervention, because we knew that if we can set up an accurate predictor with some statistical efficiency, then we could, if we could learn this by, for instance, by setting up a maximum likelihood estimator in some highly parametric system, then we could go within the system and do some analysis to see what exactly is going on, and then maybe predict some pathways. The important part here is the statistical efficiency, which means we are actually interested in setting up a machine learning problem, meaning a system that predicts outside of what it has seen, because then you know that the system internally has formulated some kind of mathematical quote-unquote understanding of the problem, and that can be distilled in some way into useful knowledge that can be reproduced experimentally. So this was the problem that we've had, and the big challenge here was that we were seeing genetic gene interventions, but we were interested in prediction potentially for some genes that were not seen in the training data, or for genes that were seen in the training data, but for which the training data had significant issues. We've had low confidence in it, or it wasn't really pointing at anything, and we had reasons to believe that the experimental evidence was not necessarily of the best quality. So this was the setup of the problem, a mainline inference problem starting from genes to some kind of a prediction, and potentially we were interested in setting up this problem for genes that we had not seen. Usually this is something that looks like this. So let's say you have a couple of genes for which you do have some evidence about their log fold counts, and then one of them for which you don't. Because you're interested in predicting for the gene for which you don't have any evidence or any credible evidence, you're interested in introducing a prior some kind of prior distribution, some kind of a metric which relates the gene that you haven't seen to the ones that you have seen. It's some form of an anchoring. And this way you can assume implicitly that there is a smooth prediction surface between the entities that you have seen, that is, because it's smooth, is also salient, and that something in between potentially has the same profile as the things that you have seen. So we were tasked with constructing the metric. Obviously, the evidence that we had before, which is the stuff from the gene expression omnibus, wasn't enough because we needed more information. So we started assembling additional evidence, additional data from the field that would be used to construct this method. And we started with the biogrid with protein-protein information because that was a preeminent data source. But then we've expanded this to as much as possible because as our methods and our understanding of this approach expanded, we've seen that we needed more and more information to calibrate the system. So we moved on to gene co-expression database, co-express.db, in paranoid sources, gene orthologs and paralogs, gene ontology terms, which are not gene themselves, but they're abstract concepts tied to genes and potentially aiding the network of genes, stitch information from proteins and small molecules, and some other sources which contributed less to our system, but they still contributed, like pathways, Myrtar-based, KEGG, and so on. So the first thing we need to notice is that we are trying to build a prior here, which is fairly general. The evidence that we're assembling here is general protein-protein interactions. They may not necessarily have anything to do with aging. And then we are biasing this towards aging, towards our mainline inference. Problem is that these sources are still subjected to experimental error or to experimental bias. So for instance, there is a lot of evidence in the biogrid regarding certain things that people found interesting, like cancer and so on, but there isn't necessarily a lot of evidence in other things. And there's also a lopsided representation of the species, which is why we focused on the three species, human mice and the C. elegans swarm. So after assembling this evidence, we've laid it out in a primary data source protocol, where each one of the nodes was either a gene or some kind of an entity from the other databases. And it was structured as a multigraph. And you can see the different types of nodes here, each one coming from a different database, and the oriented links, evidence that was coming in. Nodes could have multiple links in between them because there could be multiple pieces of evidence coming in from multiple databases. For instance, you could have two genes for which you have some protein-protein interaction evidence, and let's say some other type of evidence from the pathway database that they're part of the same pathway, right? And they were all part of this big graph, which is the primary data source. There was some database-specific normalization applied to the edges because the databases themselves had the prescriptions of how to interpret the confidence and the parameters. And then all of this was assembled into a large picture of what we knew. And this is stuff that is also expandable to other problems, and we are looking to expand it to more competent representations. But afterwards, we were interested in merging this into a single oriented graph. So this was the reduced data source, which was an actual directed graph, no more multiple links. This was essentially the step where we considered the relative confidence between the different data sources. So if you want to merge evidence from the biogrid and from co-expression databases into a single link, you need to make some kind of consideration and assumption about the relative importance of the scores that were prescribed for the two different databases. This was the quote-unquote reduced data source, which is a simple graph. And this was the stuff that we fed into the machine learning model to create the representation. And many assumptions going in here are mostly about the relative confidence of the data sources. Now, it's important to note that this is still also independent of the problem. So then we went about the task of constructing the metric. Now, normally when you have a graph, there is an apparent metric emerging, which is the shortest path distance between, now this is a weighted distance because the links are weighted. So let's say you have the distance between the red node and the empty node, and the shortest path would be a distance, of one line, which is this guy over there, between the gene ontology term and the interest term. And it isn't usually typically a good distance because it's a pseudo distance. There are some mathematical properties that are missing from this. Graphs are not really spaces. You have to actually run some more math to collapse them into spaces. But there's also a better candidate for this, which is the flow distance, which is the idea that when you have two nodes in the graph, you fill one with water, and your distance to another node in the graph is the total flow rate across the graph as water is attracted to the other node. And this may occur over the shortest path distance, but this may also occur over longer distances, some of this flow, because those are maybe important connections contributing evidence to the total flow. This is more important because it's smoother prior, and in the sense that it's more stable statistically, and it also accounts for non-local effects. So it's less likely that the shortest path distance would just be some experimental artifacts and mistakes that were made, because we're considering multiple pieces of evidence when determining what is the relative closeness of two nodes in the graph. So we've used this prior for the BP1 embedding. The BP1 embedding is the first stage of the Bucharest Pile. By the way, the Bucharest Pile is a name inspired by the Chicago Pile, which is the first artificial man-made nuclear reactor in the 30s. That's where and it's a pile because it's a stack of neural networks, each one doing one thing. The first part of the stack is this embedding, which is we're actually interested in transforming the graph into a high-dimensional Romanian metric. Now, we know for a fact that if we have a flat space, like Euclidean space, there will be some distortion, so some difference between the distances in the space and the distances on the graph. There's actually a theorem about that. And if we have negative curvature, a hyperbolic space where distance volume increases exponentially with distance, then we could potentially fit the graph into the space. But we could also overfit, because if our data is incorrect to a certain extent, then the negative curvature would capture all these errors, and we could overfit. So we have to tolerate some distortion because we're assuming noisy data, which is why we made an informed guess to keep the space flat, to make it a box of Euclidean box, not necessarily a hyperbolic one, although the literature of optimizing graphs and creating graphs embedding strongly points at negative curvature. So we've been using Node2Vec for this step, which is a deep walk algorithm, which is essentially a specialization of Word2Vec, a highly used algorithm to train embeddings in natural language processing. We assign a vector of 64 dimensions to each entity. These vectors have the property that two nodes, two entities are closer in this space if they have more flow between them in the original graph. It's an interesting training algorithm, which essentially leads to this. And as we increase the curvature, so as we make the space more spherical, we are looking at a less informed prior with more distortion. As we decrease the curvature, it's a more informed prior. And same with dimensionality, as we account for more dimensions. We can absorb more of the information in the graph with less distortion. So it's fairly flexible. It can be adapted, but we've chosen a certain dimensionality and a certain curvature experimentally. The problem here is that after this step, we obtain a distribution of nodes which is highly complex. So it looks like a very complicated 64-dimensional shape. And this is a highly informed prior, and we're not necessarily interested in that. We still need some way of making it simpler without necessarily reducing the dimensionality or increasing the curvature of the space, which would trade off too much distortion. So here we're applying the trick of approximate inference, which is we are not interested in the real solution, but we're interested in an approximation based on some prior. For instance, in variational inference, we'll have something like the factorization assumption that the distribution factorizes and so on. So we're constructing a surrogate space, which looks nicer. It's not necessarily closer to the truth, but it's about the same distance from the truth, but it's just nicer. And we're going to play with that because we don't really believe necessarily in the accuracy of the data. We've had some theoretical results on this. So we're building a variational autoencoder, which is implicit, doesn't necessarily need reparametrization. We've been applying this to the vanishing gradient problems for sigmoid activation functions. And we've published this in 2020 in the Applied Intelligence. So this is the part of the recipe for constructing the second stage of the embedding, which really crunches the representation into a smaller space. This is BP2. This is the second stage of the embedding. It is a deep neural network. It's a feed-forward neural network, and it puts all these, it simplifies all these entities into a almost uniform. Flat representations are never quite uniform because of numerical artifacts, stability of the learning algorithm, and so on. We can go as far down as the dimensionality as we want, practically to two-dimensional. Conventionally, with the Bucharest Pile, we're somewhere between 4 and 24 dimensional. The 4-dimensional representations are really agnostic, really just basic concepts. The 24-dimensional representations are better ones for our experimental optimum for good statistical efficiency for our mainline inference problem. And again, it's an instance of what they call stochastic gradient variational base, so of the variational autoencoder, but it's a specific type of autoencoder that we've developed theoretically, and it's more efficient and versatile than just simply applying the SGBB algorithm. So I just want to show you guys a picture. In the BP1, what you would see inside the representation, let's say the representation has many different dimensions, and you would see a complicated shape of all these points in a high-dimensional shape, in a high-dimensional space. That shape is like a very weird, twisted potato in many dimensions, and we're interested in simplifying that because we're interested in increasing the probability that a simple classification surface will pass between these for our problem of interest. Now, again, this is a process which has nothing to do with the aging problem that we've set up as an objective at the beginning. This is just learning representations in general in creating a very informed system which knows biology. So after Deep Walk and Node2Vec, which are conventional algorithms in literature, you get a picture like this, a very high dimensional space, 64 dimensions with a complicated object. And after the BP2 process, the relaxation process, which includes a tuple loss and surrogate losses from learning flat representations, the nodes are projected onto a lower dimensional surface and spread out to be almost uniform. But they keep the relationships between them, the distance relationships between them as much as possible. Obviously, there will be some more distortion introduced by this process, because we are introducing a very strong prior, but we continue to account for that distortion by the fact that we have noisy input data. So we're interested in a very blurry bird's eye view picture of biology because we don't really trust any particular one experiment that exists in these databases for the purpose of formulating pathways. So you get a lower dimensional box, notice the fewer dimensions in the corner, and which has the property that entities are no longer clumped up in a potato, but they're spread a little bit more uniformly across the space. This is the BP2X picture. So a simpler representation, potentially easier to classify, to draw a classification surface between them. This is the Bucharest Pile 2, again, variable number of dimensions. So general properties of this embedding, it is really not faithful to craft, but we've also constructed a less informed prior. It was motivated by the low confidence, and we found a way by using variational inference to manipulate the amount of information that we put in the prior, which is outside of the curvature and the dimensionality of the prior, which are intensely studied in obtaining graph embedding. So this is just a variational graph embedding with the ability to control, let's say, the sparsity and the distribution of the graph. So we can now use this to the given problem as an entry point, right? But it's also agnostic. It can be used for any other type of problem. For instance, anything you would like to study with the genes and so on. You can use this embedding to start off with it, especially since you'll see later the embedding not only has genes, but has all kinds of concepts in it. And there's an interesting point here. The approach that we've had in the constructing this embedding was then the general approach, the strategic approach was strongly emphasized in NLP with the development of the GPT-2 models. Now, granted, these are much larger models, but they basically start from the assumption that if you want to build a knowledge base internally, you want to set that up as a multitask learning problem. So a representation which has been developed by applying it to multiple different tasks. And even though the actual task here is the same, which is learning a big graph, because the graph was constructed from multiple different sources, which had nothing to do with each other, and these sources had behind them multiple optimization tasks, essentially the BP1 embedding, the BP embedding is a multitask learning problem, is a graph obtained by the same approach as the latest in the NLP embeddings. So how do you use this? Again, since it's agnostic, any inference problem, many types of entities in the embedding, other types of entities can be added. We have prepared Docker containers which contain all the steps from building the initial data source, then the reduced data source, and so on. There are some prescribed formats that you can use, and these data sources will be used. And then there are the training algorithms. And along with other criteria, this was used to predict gene combinations, which extended longevity. So we will present some experimental results on this later in this workshop. So that's it, I think, if there are any questions. But first, let me acknowledge the contributions to this effort. So first of all, Dr. Robi Tocutu and the Bioinformatics team for their contributions of critical importance. For the operational and logistical support, anything like machines, data scrubbing, and so on for this effort, and mostly for patiently listening to many presentations like this and filtering through the more dubious approaches and selecting the more promising ones. I would also like to thank my PhD advisor, Professor Adina Flora from UPB, for guidance through the PhD program and for supervising and advising the theoretical development support in this effort. And also Dr. Costin Chiru from UPB for his invaluable feedback, which has steered the project both strategically and as a technology specialist in applied machine learning, and for generally contributing his time towards reviewing our work. So thank you. That was it from me, and I will be standing by in the background for questions.